In accordance with the customs of this university, I open the ceremony with a prayer. Spiritus Sancti, gratia illuminet sensus et corda nostra. It means we pray that the Holy Spirit enlightens our mind and heart. Please be seated. Welcome to you all here at the Radboud University in Nijmegen, but also watching us via the live stream on Zoom. And especially welcome, and I try to pronounce it well in Nepalese, Swagotha. I hope it resembles a little bit. Welcome in Nepalese, especially for your parents, grandparents, and all your other family and cousins and friends watching us from Nepal, and for your colleagues watching us from the United States. I'm Maria van der Muizenberg. I'm a professor of health disparities and primary care, but today I have the honor to chair this specific festive ceremony for the defense of Dr. Uju. For those of you who are not familiar with the PhD defense customs in our university, I will briefly explain what's going to happen the next hour and who are present here in these nice gowns. You see besides me the supervisors, Professor Van Goor and Professor Alverdi. They have asked all their questions during the past years and discussed with the candidates, so today they will not ask questions but just proudly listen to what the candidate will tell us all. Then you see the chair of the doctoral thesis committee and the two other candidates of the doctoral thesis committee, and together they have read the thesis and assessed that it was worthwhile for defending today. And you see the other members of the doctoral thesis committee who will ask questions today, and I will introduce them when they are going to ask their question. And two of the opponents, as we call them, are joining us by Zoom. I will introduce you to them if it's their turn. But first, we will listen to the candidate who will present his work. I now give the floor to the candidate for the degree of doctor.
Thank you. With the permission of Dr. Board, and in order to obtain the degree of doctor from the Radboud University, Nijmegen, I would like to defend in public my dissertations entitled Non-Antibiotic Antivirulence Approaches to prevent anastomosis, leak, and sepsis. Even in the best hospital, in the hand of best surgeon, complications still happen following the bowel surgery. Anastomosis leak anastomotic leak is still remain 10%. It's occur following the disruption of the bowel, leading to the escape of the toxic content and cause infection and sepsis. Systemic infection and the local wound infection is still remain around 4%. And currently, we have only the antibiotic as a biological mean to treat such condition. We, under the leadership of Professor John Alverdi at the University of Chicago and Professor Harry Van Goor at Radboud University, Nijmegen, are working together for almost a decade to develop novel approaches to prevent post-operative infection. Our hypothesis is, despite major advances in infection control measures, life-threatening post-surgical infection still occurred. Their occurrence, course, and outcome is influenced by composition and function of the gut microbiome. Gut microbiome is defined as a consortium of trillions of bacteria that we harbor in our gut. And the composition and function of the gut microbiome are mainly affected by the one, antibiotic, and the other one is diet. So, studies have shown that with the plant-based, high-fiber, low-fat diet, and without taking an antibiotic, we maintain the high diversity of bacteria. But in contrast to this, if we change our diet to the Western diet, which means high-fat, low-fiber diet, and if we take antibiotic on and off, our gut bacteria diversity get reduced. it will disrupt the composition and function. And this have been associated with the very chronic health condition like obesity, diabetes. And these are the people who are having complications following surgery. So, such human clinical condition is not possible to recapitulate in the mice as they consume very healthy diet, which is plant-based, high-fiber, low-fat diet that enhance the diversity, resilience, and health-promoting function of the microbiome. Hence, in order to recapitulate the human condition in the mice, we fed them the high-fat diet, which is more typical of the Western diet, and expose them to the antibiotic. This results in depletion of the diversity of the microbiome, leading to the disruption of composition and function. And we hypothesize that the post-operative complications are determined by the health status of this microbiome. So in order to prove this hypothesis, we fed the mice 6 weeks of high-fat diet, treat them with the antibiotic just prior to the surgery as we did in our all patient, and did the 3 different surgery with the various level of risks. And then follow them on the post-operative period for the development of the complication like the lethal sepsis, anastomosis leak, and then the wound infection. So we did the major surgery, which is the partial liver resection, and the risk here is a lethal sepsis, as we all see in the patient who are admitted in the surgical ICU. Second operation we did was intestinal resection and reconnection, and the risk here is leak. And the third one is sterile back wound injury, and the risk here is bacterial wound infection. So our result demonstrate that the mice consuming the Western diet, which is high-fat diet, have increased infection-related complication as compared to the mice consuming a normal diet of chow, which is high plant-based, high-fiber, low-fat diet. You can see here in the bar diagram, the Western diet fed mouse have significant development of lethal sepsis, almost like a 60%, which is very striking. As you can see, none of the mice who are eating the chow diet, which is high-fiber diet, died because of this surgery. Similar trend is seen in the anastomosis leak surgery also. You can see here the Western diet mice develop more complication of leak as compared to the chow diet. And these mice are more likely to develop the post-operative wound infections with the various bacteria as compared to the chow diet. Interestingly, these all post-operative complication is significantly reduced once the Western fed diet mice is pre-rehabilitated with the chow diet just 2 to 7 days before surgery. Our next hypothesis is bacteria from the gut microbiome play a key and causative role in the post-operative complication. During colon surgery, due to the injury, our body release the inflammatory cues. Bacteria, which are always there, start sensing these cues, and they change their structures. They become more harmful. They produce more adherence capacity. They produce more toxin and the enzyme productions that could impair the healing and cause the bacterial-mediated leak. So in this scenario, we hypothesize that delivering phosphate carried on the mucoadhesive polymer like of polyphosphate or the PI-PEG to suppress the effect of the bacteria and prevent the anastomosis leak. So in order to prove this hypothesis, we developed the mouse model of anastomosis leak, where we did the colonic anastomosis and then inject with the harmful bacteria, and we found there is significant increase in the leak rate. In the other group of the mice, we let them to drink the water containing these phosphate compound and did the same procedure, and after injecting the bacteria, we follow up if they develop leak. And our result demonstrated that the mouse which are drinking the phosphate compound has significantly reduced leak rate. Even we infect the colon anastomosis with the bacteria, whereas the mice drinking just the plain water has significantly high leak rate. So we elucidated the mechanism that the non-observable phosphates are found to directly suppress the harmfulness of bacteria by interfering with the bacterial phosphate gene regulation. So in conclusion, modeling human postoperative complications by having mice consume the Western diet and exposing them to antibiotic enhance the incidence and severity of the most common complication following surgery, anastomosis leak, wound infection, and lethal sepsis. These complications are caused by the bacteria arising from among the gut microbiota, and use of the non-observable phosphate-rich polymers such as PPI6 and PI-PEG prevent the anastomosis leak induced by pathogenic bacteria commonly present in the gut of the patient undergoing high-risk surgery. Having presented this summary of my dissertation, I return the floor to the rector.
Thank you very much. That was very interesting. I will remember to go to eat like a mouse to stay healthy. Thank you.
Sure.
But now we're going to the serious questioning, and I would invite Professor Pickers, who was the chair of the doctoral thesis committee and who is a professor of experimental intensive care medicine here at Radboud University, to ask his question.
Thank you very much, Madam Rector. Dear candidate, it's a real pleasure for me to ask questions, because by training I'm an internist. I did internal medicine, and it's always interesting to see that you're not investigating some surgical technique or how I cut this or that, but you're actually looking into the microbiome, which is of course exactly the field that belongs to the internal medicine. And to start with the first question, I want to ask one of your paranymphs to read out loud the statement number 7.
Statement number 7. Routine assessment of the gut microbiome and its metabolic signature could be an effective strategy to predict those patients at risk for post-operative infection-related complications.
Thank you very much. So I think you took the easy way with this statement, because it's easy to say, look at the microbiome and predict the complication. Why not a strong statement on treating the microbiome and preventing them?
Honored, sir. So that's a great question. Actually, we can also do that. But what I think is treating microbiome is like, it's a huge, we even don't understand what is the compositions, like how that affect the, like what type of bacterial consortium there are and how we are going to manage them. The people are doing that. They have started doing the fecal transplant to treat the obesities as a clinical trial. So we can also do that, and that's also the one of our goal down the road to treat the microbiome if we know beforehand how it is disrupted, how the composition and function is disrupted.
All right, but don't you think that in a Western diet, that so most of these people have the wrong microbiome, and so it seems very easy to me to actually give them a high fiber and low fat diet for a while before their surgery? That would be an easy trial to conduct. Do you know how much time it takes if you are on a high fiber diet for the microbiome to change in a human?
Actually, in human, I'm not aware of. Like, it has been said that the population who are eating the Western diet has a different microbiome as compared to human. But in the mice study, definitely within the 2 days, we found the compositional changes once we feed them with the high fiber diet to the mice who are eating the Western diet. So
Yes, and actually I found something that in humans, already within 2 weeks after high fiber, that you see changes, and maybe they will become more pronounced after a couple of more weeks. Another way to change your microbiome is with antibiotics, as you said. And in the Netherlands, there is something that is called selective decontamination of the digestive tract.
yes.
We do that in the ICU because it prevents ventilator-associated pneumonia and sepsis and mortality. But this could also be done to change the microbiome and maybe have an effect on anastomosis leakage. There's one study from a Nijmegen group actually that looked at different hospitals that used or not used selective decontamination, and they found indeed that there's a difference in anastomotic leakage in patients that underwent surgery on their esophagus. So would you be able to design a study, or what would have your preference, like high fiber diet or antibiotics that selectively look at the bad pathogens, or a combination? How would you design a study?
So in order to decide the study, like definitely the antibiotic, like we need to know like the mechanism, like what is the coverage of the antibiotic. So it's not like we can kill with killing all the normal microbiome. I think that's not the ideal way to treat with the antibiotic. And basically, like the antibiotic which we are using is the broadest spectrum. It is like causing, I feel it is causing more harm to our microbiome, like killing all the normal bacteria, at the same time helping the bad bug, which is the pathogenic bacteria, growing in our gut. So that's how we are observing. So yes, as your point, selective decontamination is a really good idea to do that, but I think we still need to work more how these antibiotic, like selecting the antibiotic and what type of bacteria they are actually killing. And we are knowing that the more use of antibiotic is not the evolutionary stable strategy, because as we see, there are more growing of these resistance bacteria. There are growing more and more of resistance bacteria. So I think the best way is to preserve, rather than eliminating with the antibiotic, it's trying to build the normal consortium bacteria that's ultimately suppress the pathogenic bacteria in the gut and enhance the health.
All right. Do we have some more time?
For a short question.
For a short question. No, so I agree with you actually, and I think that we know already pretty well what kind of bacteria are killed by what kind of antibiotics, and that selective tract decontamination is exactly focusing on that, and to kill the bad ones and to maintain the good ones. So thank you very much. Actually, I'm ready. I'm very satisfied with these answers. I'm very happy that you study in internal medicine field, but I'm a little bit disappointed that you now say it's the fault of the microbiome that the leakage occurs, and it's not the surgeon's fault. So that's the backside of it. Thank you very much.
Thank you very much. We will now continue this discussion by Professor Joosten. He is a professor of mechanisms in inflammatory diseases here at Radboud University, and he's joining us by Zoom. Professor Joosten, please ask your question.
Thank you, Mrs. Rector. Dear candidate, I would like to compliment you with the thesis and also want to include your team of supervisors. It's a very interesting topic. Can we find a therapy or a treatment for the prevention of anastomotic leakage, sepsis or inflammation in patients after surgery? My first question is actually about chapter 3. Here you use an animal model to show what's the effect of Western type diet on your, let's say, antibiotic treatment, and later on also on the anastomotic, let's say, leakage due by you take a part of the liver away. My first question about your figure 1, page 52. You showed clearly that if you give mice a certain Western type diet, they gain weight, a certain amount of weight. And then you look also at the cytokines in the circulation, you measure IL-6, you measure CRP before actually performing the antibiotic treatment and the surgery. And you see slightly difference in elevation of IL-6 and CRP. And my question is, seeing this particular experiment, do you think that these data are valid, or what's your opinion about the results you showed in figure 1?
Sorry, Maha, may I know your questions once more time?
Yes, if you see the, let's say, figure 1, the results over there, that you have let's say, a not significant difference in the inflammatory markers. Do you think that you still have a big effect of the Western diet, or do you should, let's say, repeat the experiment? Because you have only 4 and 5 mice per group, and that's, I think, very limited, or not?
So, actually, I see like you're talking about the figure 1, right?
Yeah, figure 1, yeah.
So definitely here we saw that the mice which are consuming a Western diet, they definitely gain weight. And there is a significant decrease in the bacterial load, which is the DNA compositions. And there is also significant difference in the diversity of a bacteria, both in the lumen and the tissue in the mice which are consuming the Western diets as compared to the chow, which is the plant-based diet.
Yeah, but if you use more animals per group, or you repeat the experiment,
definitely,
do you expect them to see bigger differences, for example, for the inflammatory markers?
Yes, so this is we just measure like for 1 or 2 experiment about this marker, but we definitely repeated the experiment like almost like more than 10 times, and the results are really reproducible. And as you said, like if we definitely do more analysis on all those mice, definitely we'll give the statistically significant results on this.
Okay, so yeah, so that you say that you can repeat it, and it's also you reproducible data.
Yes.
And you repeat exam. Okay, and now my next question is that you also look in this particular chapter. You look at T cells and adaptive cells of the adaptive immunity to see whether they change after your invention. And you show that there is some slight reduction in the B cell. And you look at the table, and also the naive T cells, if you use the cache versus the, let's say, the wash, and you show it. My question is, why are you looking at the T cells and the B cells, cells of the adaptive immunity in this particular setup of experiments?
So our goal is to demonstrate the impact in the immunological function of these immune cells by disrupting the gut microbiome. So that's what would like to see all these T cells and B cells on the mice, which are almost like a septic and they are almost dying, and compare with the normal chow group mice.
Yeah. Okay. Because I think your let's say your total experiment with after the operation only 36 days or 40 days. and maybe it's not enough to induce changes in adaptive immune cells. But maybe you're aware of another phenomenon that is recently described. If you feed animals in a Western type diet, the most of the changes are not in the T cells or B cells, but actually in the myeloid cells, in the monocytes and macrophages. Are you aware about these studies that they focus on cardiovascular disease and obesity?
Yes.
Yeah, and do you think that when now you look at the T cells, maybe it was more wise to look at the monocytes of these animals, whether it was different, yes or no, in case of your cash or let's say, or your wash? Do you expect to see differences there or not?
So I think I really don't have I really don't have an answer of that question yet, because I haven't seen those effects, but definitely it's really a good idea or good things to track down the later for the new experiment.
Yeah, I definitely have to look at the bone marrow, because in these papers clearly show that after 6 weeks or 16 weeks of Western diet, the bone marrow is completely changed, and all the monocytes coming back in the bloodstream are hyperreactive, hyperinflammatory states. That probably contributes to also to your findings. In the let's say in the leakage over there, you have much more inflammatory processes. That's why you see higher mortality in your animals. But seeing the time, I have a small last question. Do you think that you would see different results if you have not SPF housing, where the animals are, but in the wildlife housing, but now they are reported in very nice paper that there's a huge difference in the immune system? What do you think?
So, to me, like I have done myself this experiment like almost like more than 10 times. So I have done this experiment with individual housing and also in the like a combined housing also. I did not see any difference that's really made with the housing differences.
Yeah, but that's only in SPF conditions. Eh? I'm talking now wildlife, so in the mouse that you buy in the pet shop, and there you perform experiments. Because they have completely different microbiome, and the immune system is completely different from our SPF conditioned mice that we use all we use in the animal experimental field. And I think that's not the right mice to use at the moment, seeing these big differences for the wildlife mice and our SPF mice. But maybe we discuss that later also with your supervisors. And I give the word back to the rector. Thank you very much.
Thank you very much. Then we will continue this discussion with Professor Bouvy. She is a professor of innovative surgical techniques at Maastricht University Medical Centrum and is also joining us by Zoom.
Thank you. Dear candidate, I would like to congratulate you and your supervisory team on this very important work to clarify the mechanism of anastomotic leakage. And I really appreciate it that you as a surgeon take so much effort to reveal these problems, because we as a surgeon suffer daily from these dreadful complications. So thank you for this. I was a bit disappointed to see if this work was translational. In all the chapters, starting with chapter 3, going on with chapter 4, 5, 6, 7, you only used male mice. Can you explain why you did not include the female mice?
Honored, ma'am. That's a great question. So, yeah, we really thought about using the male mice, male mice, and not using the other mice. The reason why is we want to see, we are testing our hypothesis, and we want to see like how reproducible it is. because definitely if we use other mice, like there are other factors that we need to consider and have to, which could potentially play around. Like it's definitely the female mice are like have a less inflammatory effect as compared to male. These are the factors that may play in. So we are not trying to test here between how the response goes on with the male versus female.
Okay, so you did this because you wanted to exclude some bias.
Yes.
Okay, and I heard that you are supervising a lot of honor students with their work. So, do you like to include female mice in your future work? Do you think it's important?
Yes, it's definitely important, and we must consider like the gender to test the hypothesis with like, and to check like how the female mice behave after surgery.
Yeah. Okay. So, well, as you know, we also do some animal work in Maastricht, and we indeed find that there were really big differences in outcome between female and male. And as Professor Joosten just stated, perhaps also it's interesting to include wildlife mice.
Mm-hmm.
And then another thing that was remarkable in chapter 3, the mice were just in their puberty, as your wife probably understands as a pediatrician. This really also makes a difference with the mice used in chapter 5 and 6, where they were already 12 weeks when used. So please take that into account in your further work, because
Thank you.
We have a lot of hope that you will manage to help us in our clinic. Then I was interested, one of the first papers you published on, perhaps you still know that, in 2017, were with the relation of intraperitoneal used phosphate to avoid adhesions.
Yes.
And if you want to make a relation with chapter 5, where you use phosphate intraluminally to avoid anastomotic leakage by reducing collagenase activity. What do you think is the relation? And another question is, your group in Nijmegen also showed that there were differences when you make an anastomosis high in the digestive tract or lower in the digestive tract. And in some of your work, I could not see where you made this anastomosis.
So, in this experiment, we use the anastomosis. It's a distal colonic anastomosis, which is 2 inches from the anal verge, just at the peritoneal reflection.
Yeah, so the enemas reached this anastomosis.
Yes,
Okay.
we make sure that the enema that we are giving and then the bacteria that we are infecting the anastomosis reach at the site of anastomosis.
Okay, and then if you can elaborate on the working mechanism of phosphate in intra-abdominally, intraperitoneally to avoid adhesions or to avoid anastomotic leakage?
So, based upon my previous work, which I actually did in the peritoneal adhesions, so I used the alkaline phosphatase, which is one of the enzyme that's present in our all body tissue, that is expressed in order to break down the adenosine triphosphate and to release the phosphate in the gut or in the body. So the alkaline phosphatase, that's how it has the anti-inflammatory activities, because it's get expressed, it's break down the ATP, which is have a more potent pro-inflammatory activities, and then provide the phosphate to the host or like to the cell, you know. So same mechanism here, like we as a host, whatever the microbiome, it's a bacteria or the animal that need phosphate to basically run the mechanism of, like for example, like the glycolysis, or to produce the energy, like it use even to utilize the glucose, because without glucose 6-phosphate formations, the glucose is not consumed to produce the ATP, right? So that's how, like, the abundance of the phosphates is really required to maintain the host metabolism. And similar things happen in the bacteria, like when the bacteria are under stress conditions, they actually need more phosphates, and that's how these bacteria are prone to express the phosphate acquisition gene, which is the PST mechanisms, and that consists of hundreds of other genes, like biofilm formation, collagenase formations, and motilities functions. These all genes are expressed as a form of quorum sensing. So
Okay. Just one small question. That's very interesting, your theory about this. Do you think that adhesions have something to do, adhesion formation has something to do with bacterial translocation?
yes.
Short answer, yes or no?
I think, like, based upon the results, like what we found is, like, mice are consuming the Western diet, they are more likely to develop more adhesions, and which is also related with the bacterial translocations.
Okay, thank you. I'm very glad that you do all this research. Please stay on doing this, and I like to give the word back to the rector.
Thank you.
Thank you very much. Then will we continue the discussion with Dr. De Kok. She's an assistant professor in nutrition and cancer at the Wageningen University here in the Netherlands. Dr. Kok.
Thank you very much. So, dear candidate, I also would like to congratulate you on your work and your beautiful thesis, which I think is not only clinically relevant, but also very much interesting from a nutritional as well as a microbiological perspective.
Thank you.
So I would like to discuss with you chapter 4.
Yes.
And from a nutritional perspective, I was very much interested in your diet. So you have the Western style diet, which is high in fat and low in fiber. And you compare that to the chow diet, which is high in fiber and other plant-based compounds, but low in fat. So when you see the difference in the outcomes and the mechanisms that you studied, it could be either that the fat is responsible, it could be that the fiber is responsible, and it could be that the overweight or the calories are important. So my first question is, could you please explain why you did select this multi-target diet?
Honor, madam. Thank you for the questions. So we selected this diet, as I said earlier, we want to prove the hypothesis, and we selected diet that recapitulate what we are actually eating. So what we are eating, the Western societies, they are eating like a high carbohydrate, high fat diet. If you go around, they are eating with the big potato jog with the huge Coca-Cola, and then like the chips, and then the fries, right? And they are eating all the time. So we really want to study the human disease and want to see the same effect in the mice. That's why we choose this worst diet, which is 60% high-fat diet.
Mm-hmm. But do you think, from a human perspective, that it would be feasible to switch to this chow diet? So in the mice we have the chow diet, but can we do that as human? Do you think that is feasible? And can patients do that?
I think it is feasible, and our next goal is to make those kind of chow diet, which is just like a chow diet, and do a prehab study.
Okay. So then my follow-up question is, we have the fat, we have the fiber, we have the calories. I do see why you have all these in your experiment. But which of them do you think is responsible for the successes that you see in the anastomotic healing? So is it the fat, is it the fiber, is it the calories?
So I apologize. I don't have the answer of that questions. I'm not sure, like, is that the calorie or fat or the fiber is working? But what we found is, like, if we enhance the fiber, definitely we saw there is an enhancement of the bacteria, like a bifidobacteria, lactobacillus. Those are mostly enhanced in the gut microbiome after we provide more fibers, and these are the ones that actually ferment those fibers. So those compositions are really increased, and we know that these are actually have a health promoting effect, like anti-inflammatory, basically these lactobacillus bifidobacteria. So we are thinking indirectly fiber supplement working through that mechanism, not the fiber directly working, but like it's enhancing the microbiome, which is health promoting, which is anti-inflammatory, which is promoting the healing, and also like trying to suppress the pathogenic bacteria, which are tend to make a collagenase that causes disruption of healing. So with all these factors, we saw the enhancement of healing. And I guess that's how it's working.
Okay, I think I agree on that. And I actually also would like to extend this more like from the translational perspective. So you have discussed the comparison from mice to humans, and that was also described in your thesis. So from a dietitian or a dietary or a nutritional perspective, I think there is one dietary habit of the mice that could be really important here, and that is coprophagy. Mm-hmm. So that the mice eat their own stool, which we as humans usually don't do. So do you think that this coprophagy is important in your experiments, for example, if you consider the antibiotic resistance or the pathogenicity of the bacteria?
So that's totally really good questions to consider. And we did consider about the coprophagia. So like we don't want the mice, especially to avoid the coprophagia, like we kind of strain the mice in such a way they are not allowed to eat their own poop. So that's how we design our experiment.
So how did you do that?
We put the wire bottom cases where the poop used to fall below the wire bottom, and they are not able to reach out.
Okay. So what I know from literature is that mice on these conditions have deficiencies because they lack the recirculation of specific micronutrients. Is that also something that you encountered?
We did not look for the deficiency of micronutrients in our experiment, but as these mice are feeding the high-fat diet, I guess there could be some deficient on the micronutrients too, like which we won't be able to balance.
Yeah. Okay, thank you very much. Is there time for a short question? Yes. Yeah. So I have a final short question, and that's more general question about prehabilitation for patients with a major abdominal surgery. So mostly there is a lot of interest for protein. And based on your thesis, I was interested what you think about the protein enhancement or increased protein intake before surgery.
So literature review shows that like there is a saccharolatic group of fermentations and the proteolytic. So what we eat, actually if we eat more carbohydrate, right, our microbiome is more tended toward the saccharolatic. And if we eat the protein forms, it's kind of proteolytic. And in the literature, it has been shown that the more proteolytic bacteria, they're more likely producing these amines, glycerol, P-glycerol, and then the indole formation, which are actually, I think like they are pro-inflammatory. So I'm not sure like how the protein supplement to work before in the prehab, but definitely the fiber is our main concern, and that's where we want to focus rather than the protein or the carbohydrate.
Okay, thank you very much.
Okay.
I would like to give the word back.
Thank you very much. Now I would like to give the floor to Dr. van den Berg. He's a postdoc researcher at Amsterdam University Medical Center.
Thank you very much. By the virtue of my right, but not without your permission, I would like to have a discussion with the esteemed candidate about this thesis, but not before I congratulate you and your supervisors with this wonderful research, which I think shines a completely new light on post-operative complications and how to treat and prevent them. So my first question is about chapter 4, where you describe this mouse model of an anastomotic leakage, and you give those mice, as we heard before, a high-fat diet. You gave them antibiotics, and then you performed surgery on them. So with this high-fat diet, you observed more severe leakage. So my first question is, I could not find in your data something about mortality in the mice. Did you observe this in your experiments?
Honored, sir. So in our experiment with the prehab, in the anastomosis, we observed none of the mice dying because we developed the model in such a perfect way that, you know, that like a chow feeding mice, the anastomosis healed completely. In fact, I would like to say that it's taken me 6 months to develop this model because it's so hard, because the technique is so difficult. It's very tiny that the more we do the injury at the colon, they are less likely to heal and cause the leak, and then the mice die the next day. So that's how I have faced, and with continuous development, I developed this technique where like none of the mice died. They poop out within 24 hours of the surgery. So in this experiment, we never see that the chow-fed mice having the anastomosis leak and then sepsis and dying because of just anastomosis. Definitely, like when the sepsis happen, and then we wait for a prolonged time, they just die, but not at the immediate post-operative period.
Okay, so the reason that I'm asking you this question is that these mice develop really severe leakages with abdominal abscesses. And normally, if we see this in our patients, they would be septic and probably die if left untreated.
Yes.
So how do you explain this, that after you finalize your technique, why they didn't die without treatment? And I ask you this question because I don't know if it's representative of the human disease.
So definitely the mice immune system is different than the human, right? So in the mice, like even there is a leak, they are most likely to contend and produce the abscesses that we see in our model. which is, I guess, like is representative of what we see in the clinical also, like abscesses, tiny abscesses, collections that we also see in this model. I think in the Western-fed mice, like they are more likely to cause the inflammations, and then they are more likely to develop the sepsis also, which we see in the human.
Okay. So my next question is also about this chapter. So I think if you would ask 10 surgeons what causes anastomotic leakage, at least 9 of them would say it's about the perfusion of the anastomosis, right?
Yes.
So I looked through your data and I didn't see any assessment of the perfusion of the anastomosis. Did you consider doing this? Because I'm asking the question because how do you know that the high-fat diet doesn't affect the local perfusion of the gut, and that's the reason that the anastomotic leak, and that it's not the microbes?
So we developed this model to an extent of the perfections. That's what I would say first, because like definitely if we impair the perfusions, and that could be the one possible cause of leak, which we are saying it's not the cause of leak. That could be the one. It's a technical aspect. So in order to avoid, like to prove the hypothesis, actually our hypothesis is bacteria cause the leak, right? So we want to show that without addition of bacteria and with the perfect technique, there is no leak. That's our goal. So what we did, actually we reduced the tissue trauma. Like the whole anastomosis we do at around like 8 to 10 minutes. And we never injure the tissue. Like we try to preserve the blood vessels. The paracolic artery, which we see in this mice, we never injure that. We never touch that. So if we touch or if we injure that vessels, they are more likely to leak or not heal also. That we observe, and that's what like I was frustrated about when I was trying to develop this model. And I came to know that actually we should not injure the blood vessels to reduce the perfusions, and also not traumatize the tissue too much so that it's healed. To your point, I'm not sure how the Western diet affect the perfusion. But in our model, I can say that there is no compromise of perfusions, as we don't ligate or disrupt any vessels. It's just like a simply transect the colon and just do the anastomosis. If we do that's it. In the chow-fed mice, they almost heal. Even in the chow-fed mice, if we inject the bacteria like the Serratia, Enterococcus faecalis, and the Pseudomonas, they leak. Whereas in the Western diet, without infecting these bacteria also, they leak. Because what you found is they are mostly, with the chow-fed, there is the E. faecalis that's growing on the gut microbiome. We are more collagenolithic. That means they are producing more collagenase. So we think it's a collagenase that causing that. It's a collagenolithic E. faecalis which is booming at the site of anastomosis with the injury and also with the high-fat diet that caused the leak.
Yeah, so I understand what you're trying to say in your paper and now, but how could you convince the surgeons that the perfusion of the anastomosis is okay? Could you think of an experiment to confirm the surgeons?
I think we would need a very short answer, perhaps not a complete design of a next study.
Just a technique that we also use in daily clinic more and more.
Definitely, like we have a various new iodocyanin techniques that we
exactly.
fluorescence use to see the perfusions soon after the surgery. I think we can apply in that our model to see like if that's not compromising the perfusions.
Yeah, that was the answer I
Thank
was
you.
looking for. Thank you.
Thank you very much. Then at last we will give the floor to Professor De Wild. He's a professor in surgical oncology here at Radboud University.
Thank you. Dear candidate, first of all, I'd like to congratulate you with this thesis. But I was a bit surprised reading chapter 4, because I was looking for this chapter in PubMed, and I found a completely different paper, which is called Low-Fat, High-Fiber Diet Rehabilitation Improves Anastomotic Healing via the Microbiome, an Experimental Model. So, is this ghostwriting or is this fake news? What is the paper?
It's the same paper.
Yeah, but a different title.
So
anyway, my problem was that the title from chapter 4 is different than the title from the British Journal of Surgery. So I was a little bit confused, and I see you are a little bit confused as well. So anyway, I would like to ask one of your paranyms to propose the last proposition, number 9, please.
Where is it? Surgeons who consider an anastomotic leak as an error in surgical technique have dismissed the dense and complex biology that underpins its pathogenesis. This claim continues to be perpetuated by the confirmation bias inherent in clinical practice.
Yeah, and I must confirm that perhaps I'm one of those surgeons, because I think that not, as you state, like 90% of the anastomotic leakage is a matter of perfusion, but I think at least 50% of the anastomotic leakage is a matter of surgical incompetence. And do you agree with that or not?
yes, I agree.
Oh, happy with that. Because I was expecting this answer. And one of the reasons of this answer is probably, I saw that you were trained in Nepal, and I was wondering if you have ever seen any anastomotic leakages in Nepal.
Yes, I have faced that.
Yeah, and I checked, because in Nepal, there's not a single McDonald's.
I totally agree.
Yeah, I was surprised. So there is no such thing as a Western diet in Nepal. So, and you claim in your thesis that the Western diet is one of the major causes of anastomotic leakages. But since there's no Western diet in Nepal, but you do see anastomotic leakages, there must be other things happening with these anastomotic leakages and this colorectal surgery we are performing. Can you elaborate on such factors?
Honor, sir. Yes, in Nepal, definitely we don't have this Western diet as we see in the Western society. But as we say, like, it's not just a Western diet that caused the anastomosis leak. It's a bacteria that caused the anastomosis leak. We don't know. In Nepal, there could be a patient population who is harboring the bad bacteria, like E. faecalis or Pseudomonas. And even though they are not eating a Western diet, and under stressful situations and in the age condition, these are the bacteria that could play a main important role to cause the anastomosis leak. In fact, I have dealt with that. So that's how, like, I'm here to defend the thesis, because during my early surgical career, I was applauded when I see the people having the anastomosis leak despite the good technique. And then I was wondering, like, why happening, you know? So, like, now, like, I got the answer. It could be a bacteria which are there always, which is really hard to wipe out despite the bowel cleansing and antibiotic treatments, and that could play a role And they are not eating, like even though they are there in the low level, which cause the leak when the hosts are under stress.
Yeah. And because in the last chapter, your future perspectives, you give some recommendations, which I think is very useful, that there need to be some microbiome readiness. And have you any suggestions how we can agree on this microbiome readiness for surgery? What should we change in the diet?
Well, you can give a very short one-sentence answer if you want.
So, microbiome readiness, in my thought, is like what type of bacterial consortiums the patient is harboring, especially if the patient is harboring like the Enterobacteriaceae or Proteobacters, which are most commonly pathogenic. Those patient population will land up to having a post-operative complications. In contrast to this, if the patients harbor the good bacteria, that we say Bifidobacterium or Lactobacillus, those populations will do well and have a smooth recovery after the surgery. So that's how we can think like just the microbiome consortiums and try to figure out if they are ready for the surgery. And if not, prehab with the diet that will grow up these good bacteria and do the surgery.
Thank you very much. The defense is hereby concluded. I give the floor once again to the candidate.
Having defended my dissertation to the best of my ability, I would like to thank the rector and my supervisor, as well as all those who have honored this ceremony with their presence.
The doctoral examination board retreats for its deliberations. On behalf of the Council of Deans of Radboud University Nijmegen, we have decided to award you the degree of doctor. I invite Professor Van Goor to discharge the task assigned to him.
In the name of the Lord, with the power entrusted by law to the doctorate board, I hereby confer upon you Sanjeev Kumar Ujjol, born in Banipa, the title of doctor from Radboud University Nijmegen, to which are attached all the associate legal and customary rights and duties with respect to academia and society. As proof thereof, I present you with this doctoral diploma, signed by the rector and the doctoral thesis supervisors.
Yeah. Now, Professor Alverde will speak some words of praise, the laudatio. You can be seated.
Thank you, everybody. I thank Radboud University and this illustrious group of outstanding faculty that I've enjoyed meeting for the first time. And I thank you for all your comments. They were spot on and brilliant and insightful. I just want to say a few words about Dr. Hoju. Since I've known him for more than 5 years, he came to me from Harvard and was highly recommended by Dr. Richard Hoden, who was a principal investigator in a laboratory there. And he told me that Dr. Hoju was one of the best postdoctoral fellows he had ever worked with in his 20 years at Harvard. And he said, could you please consider giving him a position there, since his wife was moving to Chicago and was taking a position on as a neonatology fellow. And I took Sanjeev in, and as I mentioned to the group, at one point, he worked so hard on Saturdays and Sundays, Saturday night, Sunday night, that I got a bill once for mice of $10,000 in 1 month. I said, Sanjeev, slow down, okay? His enthusiasm, his passion, his excitement, he would come to my office and I would say, you know, we really ought to look at that. He goes, I've done it. Here are the data. I go, when did you do this? And he said, well, last weekend. I stayed here from Friday all the way till Sunday night.
Oh.
So these are the kinds of characteristics when people talk about obsession and passion and commitment and engagement in a project. This is the kind of enthusiasm. And I think it resonated with everybody on this board, Sanjeev. In our discussions, in our deliberations, passion and enthusiasm and commitment were something that we all saw and heard from you today. So I want to personally congratulate you and your wife for such an outstanding presentation. I want to thank Dr. Van Goor for his collegiality. We've been working together for almost 10 years, and I got to know him when he was president of the Surgical Infection Society Europe, and it's been an outstanding collaboration. And all of you, thank you. I see many of the students that he sent to my lab in the audience today, and it's a great pleasure to have you all here and to be in this outstanding university with such outstanding scientists, and you are now one of them, and you should be very proud of yourself.
Thank you.
Dear Dr. Hoju, it's an honor and a pleasure to be able to congratulate you on behalf of the Council of Deans. You did an excellent job. It was very interesting for me to learn lots about all these food things and other stuff. I hope you will continue your studies, but I'm sure you will do so, as we have heard from your supervisors. And I would like to extend my congratulations to your partner and your parents and grandparents, brother and other family who are listening, I hope, by live stream, and your family and friends. I know, well, it's a pity they cannot join us here, but I'm sure they will be very proud of you. Before we conclude this meeting, I would also like to give the opponents who have joined us by Zoom the opportunity to give them your congratulations. So, Professor Joost, if you would like to say something.
So, dear candidate, dear Dr. Hoju, congratulate you. This was really well deserved, and I hope you continue working in this field and be involved in many of these very interesting papers. Again, very nice, deserved, and congratulations.
Thank you.
Thank you very much. Professor Bouvy?
Sanjeev, congratulations.
Thank you.
You do great work and really help us to be better performers as surgeons. Thank you also for hosting Audrey, Jonge, and Valerie. They also loved the way you supervised them. And I hope in the future I can still keep on sending students to your lab and that you will help them to become as great as you and your supervisors. Good luck.
Thank you.
Thank you very much. I would like to thank you all present here or on Zoom for your attendance. We've now reached the end of this defense meeting, and in the customs of our university, we will stand and conclude with prayer. Gratias tibi agimus, omnipotens Deus, pro omnibus beneficius tuis, qui vivis et regnas per omnia saecula saeculorum. Almighty God, who lives and reigns forever and ever, we thank you for all your benefactions.